delay, né? Vamos esperar um pouquinho.
Tá ao vivo já.
Então, good afternoon. We are back for the very last public lecture of the symposium. I am Ângelo Pinto from the graduate program in entomology. And this event is part of our internationalization program, Print Caps of Federal University of Paraná. Again, Dr. Pedro Dias will be hosting the talk. Pedro, your turn. Thank you so much, Márcio. Bye.
Thank you, Angelo. I'd like to introduce you guys to Dr. Márcio Pie. He's a professor here at the Universidade Federal do Paraná. It's really nice to have him here talking. He's my supervisor now in the postdoc, and he's going to talk about his finds in the field of biogeography. I hope you enjoyed the talk. Thank you very much, Márcio. Now it's with you. Thank you.
Thank you, Pedro. First of all, I'd like to thank you all for joining us for this discussion. I'd like to thank also the organizers for inviting me. It's been an honor to be here and share some of the work that we've been doing in our research group for the past few years. But before I start, I'd like to acknowledge some of the institutions that have supported us over the years in terms of funding and other types of support. So the reason why we decided to study biodiversity, the reason why we decided to become biologists, probably has something to do with the diversity of life and how intricate and varied it is across different types of biomes, different types of environments, and so on. But one of the intriguing aspects of biodiversity is that it's not continuously distributed across the earth. There are some regions that have disproportionately larger number of species, and this has been a conundrum for biologists and biogeographers and natural historians for centuries. But it's only recently people have framed this in terms of the three mechanisms that are actually responsible for generating species richness gradients, which are first speciation, the generation of new species, extinction, which is the loss of species, and finally changes in the geographical distributions in size, in terms of the geographical size of the geographical distribution, the shape and the position of the distribution across landscapes. So one of the things that I wanted to talk to you guys today is whether there are general principles governing the geographical range evolution. And this is important because, for instance, based on work such as the work of Dr. Sammartini and other people that are working on biogeographical problems, there's, for many people, there's a sense that biogeography is mostly idiosyncratic, basically random events that are breaking up and causing colonization of different lineages in a sort of haphazard way. But we know that there are general patterns governing geographical distributions of many lineages. For instance, we know that most species tend to have relatively smaller geographical ranges. And also if we put the x-axis here on a logarithmic scale, we tend to have something like this, where we have an approximately normal distribution, or in this case, log normal distribution with a left skew. So this is something that we've known for quite a while, and this is very general for many types of organisms, from plants to mammals. But in a sense, when we look at the geographical at a given geographical distribution, one thing that people might not have in mind is that we're assuming that there is such a thing as a geographical distribution. We assume that one monolithic thing known as a geographical distribution exists. And we try to infer the mechanisms driving its evolution. But one thing that we could wonder is, what if different regions of geographical distributions evolve according to different rules? For instance, this is a work that I did with a former PhD student in my lab, Andrés Maia, which is the mammal here on the right. And this is an example just to give an idea of what we did. So this is a star-nosed mole, and it has a distribution all across from Newfoundland here all the way to Florida. And if you look at this distribution, it becomes obvious to anyone who knows a little bit about the climate and the ecological conditions in this region, which is the factors that limit the northern distribution of the species are probably drastically different from the factors that affect the southern distribution. So one thing that we did was to take the northernmost point in terms of latitude and the southernmost point in terms of longitude, and we considered this the high latitude limit, which is the limit closest to the poles, and the low latitude limit, which is the limit closer to the equator. And one thing that we wanted to ask is whether these limits evolve according to similar rates or different rates. And this is just to give you an example of the type of data set we use. We use thousands of species of mammals here on top and reptiles here on the bottom. And what we see over and over again is invariably the limit closer to the poles, or the high latitude limit, it evolves substantially faster than the limit closer to the equator. So in each one of these three graphs, bar plots, we have the lowest latitude, the midpoint, and the highest latitude of different lineages. And here we have artiodactyls, carnivora, bats, marsupials, and primates. And here different types of reptiles: Angomorpha, Geckoda, Iguanida, Lacertida, Syncodia, and Serpentes. So basically what we have is not only it evolves faster, but 3 to 4 times faster than the low latitude limit. So really shows that the high latitude limit evolves at a completely different tempo than the low latitude limits. And one thing that we've been doing more recently is to expand this type of analysis to other realms, like here in the context of reef fishes. This is a work that we just submitted, including a few colleagues, Brazilian colleagues that are currently based in Australia, Osmar Luiz, Alexandre Siqueira, and Diego Barnesch, and also 2 students in my lab, Raquel Divieso and Fernanda Caron. So basically we also compiled a really large-scale data set of reef fishes. And we found all sorts of really cool things. For instance, we found that in terms of latitudinal range, they also share the same pattern as terrestrial taxa, in the sense that they have an approximately log-normal distribution with a left tail. We found, for instance, that they tend to have what we call a reverse Rappaport's rule, in the sense that the ranges actually become larger as they come closer to the equator. But most importantly in our context is that they also much more often than not, the high latitude limit also evolved much faster than the low latitude limit, which shows that this is a trend that is constant both on land and in the ocean. Another project that we've been working on is to try to look at whether different biological characteristics might affect the rate of evolution of geographical ranges. In this case, we looked at homeothermy, which is a very important physiological trait for terrestrial vertebrates. And this also has been done with this very bright students in my lab, Raquel Divias and Fernando Caron. So, for instance, if you look at the distribution of range sizes for both ecto and endotherms, you tend to see something like this. So the red line is the mean, and we have a distribution of range sizes that's approximately log normal. And just to give an idea of why there's this truncation on the right side, this is the size of continents. So basically, the ranges can get much larger than this because of geometrical constraints in a sense. But when we separate this into different clades, we see some really intriguing patterns. For instance, if we look at reptiles, they tend to have relatively smaller geographical ranges. And also, if you look at the left part of the x-axis, there's an abundance of species with relatively small geographical ranges. And we see the same pattern here also for amphibians. We have a very high frequency of small ranged species and the overall relatively small range size. But when we look at mammals, for instance, we see two really clear differences. One is that the ranges become much larger across all the groups that we studied, and also we noticed that there's a deficiency, or there's a scarcity of small ranges, except for a little bit here in primates, but in general, we don't see the small ranges that you see in ectotherms. And this is also shared with birds, which also have larger ranges and relatively few small range species. And we discuss this in more detail in the paper, and we can send you a preprint if you're interested. But also there's this interesting pattern here in snakes, which tend to be sort of halfway between what you see in endo and ectotherms. And when you look at within snakes, you see that the pattern is mostly concentrated in the elapids, to some extent in viperids. Colobroidia tend to share similar patterns as other ectotherms. But here in elapids, they really tend to show an endotherm-like pattern of range size evolution. And we're still trying to figure out why this is so different from other ectotherm groups. And this is an analysis that we did across thousands of species, and we looked at hundreds of different 1,000 different alternative topologies for each of the clades. And that's why we have these confidence intervals. But the take-home message from this is that not only endotherms have larger range sizes, but they also evolve much faster, both in terms of the size of the range and the latitudinal position. So in a sense, when vertebrates became endotherms, it allows them to adapt and to change their geographical ranges much faster, both in terms of their size and in between different regions. So it really looks like the evolution of endothermy marked the fundamental shift in the way that endotherms evolved their geographical ranges. So I want to shift gears a little bit here to focus on one very important concept in ecology, which is the ecological niche. And since the early formulations of the concept of Hutchinson's multidimensional n-dimensional hyperspace, hypervolume as a metaphor for the ecological niche, even in Hutchinson's original paper, there was this duality in the sense that you have species or different lineages occupying different regions of niche space. And this would show consequences for the way that these species are distributed in geographical space. But one thing that has been missing from this point of view is that we know relatively little in terms of how the actual ecological niches evolved and how it translates into how species are distributed. And so basically, what we can try to figure out is how does the niche width change and how that evolves across different types of lineages. So just to give an example of what I have in mind here, we have temperature axis, and we can imagine an ancestral species that can evolve its ecological niche. So basically, it can evolve by, for instance, shifting the position of the niche across the axis without changing its width, or it could change the breadth of its niche. But this change doesn't have to be symmetric. For instance, you can have asymmetric changes in the upper and lower limits of the range. So basically, there's many ways in which you can have an increase and decrease in the ecological niche. And we wanted to explore this in terms of their climatic niches. So I'm going to show you a couple of diagrams using this sort of formula. Basically, this midpoint is the average temperature across the range of different species. Here is the average precipitation across different, here we have 2 species. And so this is the average precipitation, the lowest precipitation, the highest precipitation, the same here, the lowest temperature and the highest temperature for these species. So basically we did this for thousands of species or different clades. And this is what we see. So here we have birds, mammals, squamates, and neurons. And you can see from this plot alone is that it becomes clear that these lineages tend not to move around a lot in terms of their upper temperature limits, but they change a lot in terms of the lowest temperature climatic niche width. But the opposite is true for precipitation. So basically, the highest limit in terms of precipitation of a species' climatic niche tends to vary considerably faster than the lowest limit. And this applies to all these groups, regardless of whether they're endo or ectotherms. And when we look at the rate of evolution in terms of the parameter estimated for the rate for each of the groups, we see something that's analogous to what we see in terms of the high latitude and low latitude geographical limits. We tend to see the lowest temperature limit tends to evolve considerably faster than the highest latitude limit. And this is the midpoint, so the average condition across the range of the species. So basically, this is similar to what we saw in terms of the latitudinal limits, but here specifically in terms of temperature. But on the other hand, we see something qualitatively different in terms of precipitation. Basically, different lineages evolve very little in terms of their lower limit and the high limit of their distribution. Basically, you have different limits. These boundary conditions are relatively constant, and the species changes sort of the degree of skew in one direction or the other, but without changing the envelope, the total envelope of precipitation niche. And this pattern, this graph shows this a little bit more detail. So basically, we have on the x-axis, we have mean annual temperature, mean annual precipitation, and here we have niche breadth. So the extent of which, for a given species, you have more or less temperature across the range of the species. And what we see is that as you get harder, the width of the niche breadth tends to decrease. And this is true also across all of these organisms. So amphibians and birds, mammals and reptiles, they all share the same property. On the other hand, we have a positive relationship between niche breadth and precipitation. So basically, on the left-hand side, we have species that are adapted to very dry conditions, but dry conditions all year round. They have very specific, very specialized niches. On the other hand, as you get further to the right, you get species that have much broader precipitation niche breadth, in the sense that on one season they can be in a condition of very dry conditions, but on the other season they can be subject to very high precipitation, for instance. And one hypothesis that we tested is whether species that are they have broader niche in one axis might have a narrower niche on the other axis, sort of a trade-off between niche breadth in one axis as opposed to the other, which is a relatively old idea in niche evolution in ecology. So basically, we did find a negative relationship, but the R squared or the strength of this relationship was relatively weak. But this got us thinking, why would these organisms, they have vastly different ecologies and vastly different geographical distributions and histories and everything, why would they share such an obvious pattern in terms of their niche breadth evolution? So one thing that we did was, what if this is actually the way that climate is distributed? What if it doesn't have to? What if it's not related to the way species are adapted to different conditions, but to the way that the conditions are actually distributed across space? And this is what I did here. What we did here was just to take the same climatic conditions, and we just did a grid of the entire world for as if it were each cell was a different species, and we got exactly the same results. So if you compare this with this plot with the previous ones, basically we see exactly the same pattern. So in a sense, this shows us that species are not evolving particular conditions or particular ways of organizing their climatic niches. Species are just adapting to whatever combinations of climatic conditions are present across their range. And this is an idea that we also explored before. So for instance, when you look at the original plot showing the climatic niche conditions in Hutchinson's n-dimensional hypervolume analogy for the climatic niche, if you look at this plot, there's no obvious reason to expect that there are limits to each of these niche axes. So it gives you an idea that niches are distributed homogeneously across a niche space. And even if you look at a three-dimensional or an n-dimensional space, it implicitly assumes that all climatic conditions or all combinations of climatic or niche conditions would be equally likely. But this is definitely not true. So this is a study that we published a few years ago, in which we calculated how much area is there for different climatic conditions, in this example across the neotropics or South America. So basically, we binned the climatic conditions in terms of different climatic brackets. For instance, from -8 to -6, -6 to -4, and so on. And we calculated the area for each of these climatic conditions. And we get a graph that looks a little bit like this one. So in a sense, what we have is that there's a lot more area in which temperatures tend to be relatively warm. So between, say, 24 and 28 degrees on average across the year, or in terms of mean annual temperatures. And what we showed in this paper is that not only species tend to be found more commonly in the climatic regimes that are more common. So in a sense, we introduced the idea of environmental prevalence, which is the area, the geographical area that contains a given climatic condition. So basically, species tend to evolve and to adapt to whatever condition is more common. So in a sense, it's not unexpected to see relatively few species under these conditions, not because these climates are particularly inhospitable, but mostly because these climates are rare. So in a sense, species are adapting to to the conditions in proportion to their prevalence. So a few take-home messages from what we saw today. First of all, we saw that there actually are general principles governing the evolution of geographical range sizes. So we showed that, for instance, different regions of the geographical range tend to evolve according to different rules. The northernmost limit or the limit closest to the poles tend to evolve substantially faster than the limit closer to the tropic, to the equator. And this seems to hold for regardless of whether you're talking about vertebrates or ectotherms or endotherms or land mammals or land animals or ocean animals. We still haven't tested this for plants or other invertebrates, but this should be a very interesting area for future research. Second, we showed that geographical distributions evolve substantially faster, almost twice as fast in endotherms, both for geographical range and position. So this tells us that the speed at which the geographical range of a given lineage evolves depends fundamentally on their biological characteristics. So in a sense, by studying these traits across different species, we can probably figure out important consequences for their biogeographical distributions, for different patterns of endemism, and for their capacity, for instance, to overcome dispersal barriers, for instance. And finally, and this is something that has been that we've been exploring for across all these papers, and this is something that sort of permeated all of these analyses that we've done, that we showed you today, which is that lineages are not evolving different conditions in the vacuum or different positions, independent positions across their niche space or their ecological space. It seems that lineages are simply evolving and adapting to the template that's provided by the conditions that they find in nature. So in the sense, by what we're calling the non-isotropic nature of climatic niches, or the fact that different combinations of climatic conditions are relatively rare and other are common. This is something that drives the evolution of climatic niches and the distribution of species, not the other way around. And so this has important consequences for understanding their distributions. And this is something that I've always found a bit frustrating about many ideas in biogeography, because something that I mentioned before, oftentimes biogeography looks like a whole bunch of accidents that happened, or species that happen to cross a different dispersal barrier and a different vicariant barriers that split up the distribution of different species. So it always sounded a bit opportunistic, in a sense, a bit stochastic. But what we're trying to show with these results is that there are underlying patterns that allow us not only to understand the distribution of species, but also to predict how they're going to evolve according to their biological characteristics. And this is what I had to present to you guys, and I'm happy to answer any questions.
Thank you, Márcio. Thank you a lot for this really, really interesting talk. Now I would like to invite all the participants to join us in Microsoft Teams. Márcio gonna be there to ask your questions and answer all of your doubts. So thank you once again, Márcio. See you on Microsoft Teams.
All right.